Let's cover a few basics of measuring biodiversity that we can use in more specific discussion about how to do it. So this is some of the basics. You may have had some of this in some of your other coursework, and let's just run through it so that we can move on to the more kind of tricky things. All right, so measuring and comparing species diversity or community composition. There are a few basic things that we want to keep in mind. Some terminology here. The first one, I'm going to talk about the species evenness. So this is in counting for not just who is there, but also how they are distributed. So their distribution in terms of their abundance and the number of individuals per species. This is a good thing to measure in a system because it accounts for rareness, which may be something of specific interest to you when you're trying to maintain biodiversity. Importantly, you can have the same richness within an ecosystem, but different evenness. And depending on what you're trying to do, this may be important to you. So let's look at a system where you have similar richness, but different evenness. And that would be a Pinyon pine ecosystem. This one, here's the Providence Mountains, and then a Sierra High Meadow. This one's on the Miriam Plateau, or excuse me, the Humphreys Plateau in the basin, Humphreys Basin in the Sierra Nevada. You can have similar levels of biodiversity, but different distributions here. And like I say, depending on what you're trying to do, this may be important to you, or when you are trying to maintain or quantify biodiversity, especially across the landscape, these two kinds of ecosystems may have different values. And you may find that each one of them has greater importance depending on what you are trying to do. Okay. Individual communities have a number of characteristics that we can point to and that you can quantify. Species richness is probably the simplest, and this is just simply the number of species that are present. One thing to keep in mind about richness is that detecting rare species is hard. It takes more effort. You need to sample lots of locations or lots of area to do it. And what we find is that usually because of differences in evenness within systems, the most common species you find in your first few sampling points. But then from there, things get a little bit harder to quantify. You got to have a lot of plots if you want to find the rare species. There's also a difference between simply species and their function within ecosystems. Individual species have different levels of importance. And so some rare species might be important for distributing seeds, or species that might be especially gigantic and create the primary structure of an ecosystem, but be less common. Or you can have things like a mycorrhizal fungi, which may not have the most biomass, but are critical for maintaining biodiversity and so forth. And along those lines, things like shade tolerance can also be very important in creating and maintaining biodiversity. So as mentioned, the more you sample, the more you find. And what is generally happens is that usually the more you sample, you will eventually start to reach a plateau in terms of the number of species that you are quantifying. Here's an example of a fairly low biodiversity system, an agricultural setting. It doesn't take much effort, and most of the species have been found here. Contrast that with a very hyperdiverse tropical rainforest where it is difficult to quantify biodiversity, and you may not ever reach the maximum amount. These kinds of relationships occur in many different kinds of ecosystems. This is the wildest one that I can find. This is a sampling of microbes in the Palmer Peninsula, or near the Palmer Peninsula in Antarctica. And what you've got here is that as these islands, you've got these islands have microbes on them. They live on these little rocks and stuff, and they're there, but they're rare. And the more you sample, the more you find. So that's just another example of how this is consistent across different kinds of systems. And if you were trying to do a good analysis of biodiversity on the Palmer Peninsula, you would also probably need to sample more on the main land mass than on the smaller islands, because as we know from theory of biogeography, the smaller islands should be less diverse and the mainland should be more diverse.